Over 44 million individuals are afflicted with Alzheimer's, the most deadly neural disorder worldwide. Scientists believe that a buildup of the corrupted amyloid, an essential protein in neural growth and repair, is the leading risk factor for Alzheimer's. However, the extensive array of treatments and billions of dollars into research and development into amyloid has produced little to no advancement for this disorder. Not much testing has been performed outside the space, with only 10% of the grants going to genomics due to the complex neural system and lack of genomic understanding from these researchers. Hundreds of gene and RNA targets are not yet explored, each of which can make an instrumental impact inside of the drug discovery process. Currently, financial investments are waning from public and private investors due to a lack of progress in the space. 60 to 70% of the investment comes from the industry, meaning that funding as a whole for neurodegenerative research is declining. By using genomic data and MRI imaging, we aim to discover possible gene targets through a 6-step process for future drug discovery methods in Alzheimer's and other neurodegenerative diseases detected inside of brain scans. First, we detect different regions of the brain using a convolutional neural network image segmentation model through pre-processed MRI imaging data inputs. Then we utilize an artificial neural network to map these inflamed regions along with these levels of inflammation. Next, scatter plots for these gene expression for various brain regions are then superimposed to see whether these genes are overexpressed or underexpressed that can further cause inflammation, and then we compare this to genetics from these inflamed regions. Afterwards, the TSNE feature selection model can then be used to visualize this high-dimensional gene expression data into more readable and visualized two-dimensional format like the one you see in the picture above. This can be seen to find more specific gene variants that can be used for differential gene and transcript expression that can find statistically significant gene features for targeting specific genes for neurological diseases, and we use frameworks like DE-seq2 and Sleuth. Afterwards, through mapping and comparative analysis of these different inflammatory gene expression levels against a neurodegenerative expression profile, we found 4 genes that had the same expression level: GRB2, MAPK1, PRKCG, and IL1B. The same trend showed true for the average expression levels for each gene that was relative to the area. Specifically inside of the hippocampal formation area, there was a strong correlation with clustered genes and the ground truth, indicating that there was an early onset of gene expression in this region that directly correlates to what Alzheimer's disease produces. And note that we did this research for all the regions of the brain, so this insight can be found for each of its functions. Also, note that when feature selection was performed by TSNE, we discovered that this gene expression was dissimilar throughout the different regions of the brain, showing that the uniqueness of this correlation can only be seen inside the hippocampus area. With our workflow and results, clinical researchers can run testing with these modifications of these different things through gene editing to see how it actually affects brain function. Then this open source workflow can also be interpretable in terms of other neurodegenerative diseases as well, such as Parkinson's, allowing for more affordability and accessibility. Our team is excited to take this project further through the integration of MRI scans and also just applications in other places of neurodegenerative diseases. Thank you.